Okay, so thank you so much, Wolfgang, for this very nice introduction. It's a real pleasure for me to be here. Yeah, and it was really fun to hear how, in the previous talk, how, flexible electronics has come to. I remember in '94 when I was at Bell Labs and we started working on organic transistors, and when we were talking about flexible and rollable displays, people was watching at us saying, well, that sounds really strange. It's not strange anymore. So it took 25 years though. Anyhow, I'll switch to working on electronic biosensors, and I will be talking about this journey we have been doing since the last 10 years, I would say, working on making those systems very, effective and also reliable. And here we go. First things first. So the team, this is really a team composed by a number of people. I'd like to acknowledge contribution from the University of Bari, particularly Dr. Eleonora Macchia and Professor Gaetano Scamarcio. And then the team at the University of Brescia, Professor Fabrizio Torricelli, who is also chairing a session here, and Professor Ron Ostebacher, who is also chairing a session here. And I will be talking about work that we did in collaboration, and in fact Professor Torricelli is also co-leading some of those efforts. And then many other partners, particularly of the European project, that work with us on those on those work, and companies. And here there is the CEO also FlexEnable, with whom we are working in the Sydney project. So I will be introducing you what we have baptized as the single molecule with a large transistor technology is called CMOT, and it basically conceptualizes cell ability to track at the single molecule level. Why it is important? Well, because it allows you to get with a large device, in fact, a printable device, the lowest possible sensitivity, and eventually those systems may allow for early detection in point-of-care devices. And then I will be just giving you a glance of what was there before as far as label-free single molecule detection before 2018 when we published our first work. Then I will be talking about presenting you some of the projects we are working on, and this will come. And I will give you also a very brief description on how our system works. And perspective, among the others, a center of single molecule that will be funded by our local government, the Regione Puglia in Italy. And in fact, I'd like also to advertise here that our local government offers very, very convenient conditions for companies that wants to put something in our region. And then the SIMBIT technology, while the bioscreen was just, one single device, the SIMBIT technology is about developing smart sensors into an array, an ELISA plate that is all electronics, and it's able to detect at the single molecule level. And this is how the SIMBIT system looks right now. In fact, it's a bit more advanced than this, but clearly, and this is the ELISA plate, and this is how the SIMBIT final device would look like. So an ELISA plate where there is all an electronic system and where all electronic detection can be performed. So, and this is basically the SIMBIT consortium on the Lake Garda, and Basically, what we are aiming at is world record detection in protein sensitivity. It will be the sole system to assay both genic and genic strands and proteins. It will be super fast, cost-effective, and multi-purpose, offering also multiplexing. But why is this important? Well, basically, we are working at technologies that will endow the clinicians with the advantage attacker, basically on the disease, on the developing disease. So the sooner you spot markers into hopefully peripheral biofluids such as saliva, blood, or even saliva, well, if you do so, you give the doctor the chance really to make a diagnosis very, very early and enhancing chances for the cure. But what's the concept here? As I was anticipating, we apprehended that cells can track a single chemoattractant or even a photon at their surface. But cells are not nanometric objects at all, meaning that the footprint of the protein or the chemoattractant that the cell is detecting, it's up to 10 orders of magnitude smaller than the wall surface of a cell. So there's no way it's the area footprint that is really at the basis of the transduction mechanism. There has to be an amplification step there, and while the trafficking of the cells that can detect a single molecule, the cell trafficking can be very, very different, they are all endowed by one specific properties. Their surface is covered by an extraordinarily large number of chemoreceptors that are highly chemoreceptors, yes, that are highly packed. And so this is the feature we wanted to implement on the gate of our transistor, and with a very much optimized procedure, we were actually able to endow a centimeter large gate of a transistor with millions, we attached to it millions of antibodies, trillions actually of antibodies. And this allowed us to see a single molecule of IgG while we were completely blind to other molecules such as immunoglobulin M. And this is a general, sketch on what the device looks like. The important thing is basically that the detecting interface is extremely large, is half a centimeter in area. And the transduction mechanism is basically a change in the work function of the device when the analyte, the immunoglobulin G in this case, selectively interact with the receptor, with its receptor, and this basically shifts the work function, changes the work function, which shifts the threshold voltage of the device. And again, this was published in 2018, and here I'm not going into too much of the details, but just to show you, this is the calibration curve. Oh, sorry. Let me do this. And this is the calibration curve of single molecule detection, and how we managed to decide that this is actually single molecule. I'll show you just in the following slide. And so we can detect IgG in PBS, in saliva, and in whole blood. And now, what's the difference here between what was already out there in the literature before our work? Well, single molecule label-free assay was carried out with nanometric interface. It's clear that at this point, the footprint of the protein to be detected and the detecting surface is comparable in size. This is very nice work, but this is single molecule resolution, not single molecule detection limit, has the concentration that can be assayed are extremely high, let's say nanomolar and micromolar. Because this is so, because basically if you have a nanometric interface, the cross-section between the nanometric interface and the antigen, the protein to be detected, it's high only if the two are restrained in a very small volume, let's say 1 femtoliter. But 1 femtoliter is by no means, volume you want to process in a real sensing assay. When you do a real sensing assay, you want to go to 100 microliters. Let's say you want to shrink it down, you can go down to 10 microliters, but it's not feasible to handle volumes of fluid that are smaller than this. So 1 femtoliter is basically 1 over 10 to the 11th of the volume, which basically means that if you want to feasibly detect a single biomarker with a nanometric interface, you need to have one of your antigens or protein to be detected in each femtoliter that composes your 100 microliter volume, and this basically means that you are working with a nanomolar solution. This is basically called the diffusion barrier issue, and if I'll have time I'll show you that when you have a large interface in 100 microliters, this issue does not hold anymore. And this is a great advantage for a technology that uses a very large detecting interface to do single molecule. And in fact, this is exactly what we did. We basically took a large interface, we conceptualized, we learned from cells that are able to actually capture single chemoattractants at their large interface, and we developed our device actually being able to sense down to the zeptomolar level, tens of zeptomolar, that in 100 microliters is basically one molecule. But this sounds really strange because we have a factor of 10 to the 12 now between the footprint of the protein we are detecting and the actual detecting interface. It's like spotting a single droplet of water in a lake or a pond, which is 1 kilometer wide. So nonetheless, this is possible, and now we are working at optimizing this sort of technology. But what is out there commercially wise? Well, in biochemical detection, well, there is the workhorse since 40 years now, the ELISA that detects proteins. Then there is the real-time PCR and the next generation sequencing, which are very sensitive, but they are restrained to detecting to do amino acid detection. And then there is the Simoa. This is a new technology that detects proteins actually at the attomolar level. And here we go with what they are able to do. So ELISA goes picomolar, while real-time PCR and next-generation sequencing, they can go down to a single copy of a DNA strand, but they cannot detect proteins. And as to proteins is concerned, we can go down to attomolar, maybe subattomolar, but there is still a range between 10 zeptomolar and attomolar, which is not covered by any commercial technology, and this is where the SIMBIT technology can squeeze in. And now we have a Simoa planar array in our lab in order to benchmark, our data. And as I was telling you, usually when you do a detection and you want to have a given
you want to have an idea of what will be the errors that will affect your technology, you basically you have to, study what is the noise level. We do so by doing a negative control experiment, IgM, that interacts with our anti-IgG interface. We took the level of the noise, the average level of the noise, we add 3 times the standard deviation, and this is the limit of detection that basically gives you 1% of false positive, but up to 50% of false negative. Nonetheless, the limit of detection falls right where we have one single protein. We were able to detect with this technology a single protein and a single nucleic acid in the SYMBIT project, where we are basically, the aim is to develop a technology that allows us to do a single early detection of pancreatic cancer, and by looking, and now this is done by very, very invasive techniques, and now we want to do it with our CMOT device directly in blood, and so we were able to detect KRAS and MAC1, which is known to be markers for a pancreatic cyst, and able to say if a pancreatic cyst will develop into a malignant one, being likely a precursor for the pancreatic cancer. And again, with a single technology, we were able to detect both genic and protein markers. And here we go, I'll show you those in blood. Again, extremely low detection limits that were possible with our device. This is a single device, and clearly in the SYMBIT project we are working in making this an array. Well, we are working on the mechanism, and the idea basically is the following. We have a layer of capturing antibodies on a large interface. There is one capturing antibody which is there and interact with a single marker, IgG in this case. This is an electrostatic. This causes electrostatic changes in the molecules because it's an allosteric process, and the energy that is generated, the electrostatic disorder that is generated there basically affects only one capturing antibody. But the idea here, and so this is basically the surface potential that changes before the binding and after the binding, but due to collaborative electrostatic interaction of the capturing antibody in the capturing layer under the electric field that we use to measure our device, basically we have a spreading of the electrostatic change. It's like a domino effect, but now those are the tails. If they are close enough, if you strike one, all the other will fall, provided that they are close enough. And while this process is fueled by the gravity field, in our case, the process is fueled by the electrostatic field. And this is a simulation that says how, the dipole moments that connects the proteins into the capturing proteins look in the electric field. There is an hydrogen bonding that basically acts as a collective system, and when you just take away one or two single hydrogen bondings there, and you put your sample in the electric field, the electrostatic change does not involve just, the molecule or the single dipole that is involved, but it spreads, changing completely the landscape of the surface potential of the gate. And I guess I have also 1 minute to talk about why the Brownian diffusion is not really an issue in our case. Well, the point is the following. If you consider Einstein equation for the diffusion, and you consider, your IgG antigen that is close enough to your interface, detecting interface, and you consider the whole volume, let's say 100 microliters, and you make the probability that, your IgG will be close enough to your antigen is given by the chances that one IgG falls in the volume that is within the diffusion length of your molecule of your antigen divided by the whole volume. And under the assumption that at least one antigen out of 10, for instance, that are in your 100 microliter volumes strikes your interface, giving rise to the amplification process I told you about. So this is basically the equation. I'm not going into the details that models what I just described. And under this hypothesis, the probability function we built, basically, it's able to model our data in 100 microliters volume and in a larger volume, showing that it is actually feasible that the diffusion limiting effect that actually is plaguing nanometric detecting interface is not plaguing our devices. So the take-home messages are the following. I hope I convince you that the SYMBIT technology is really able to comply with all the requirements of the reassured, system that was brought up by the World Health Organization to describe what is needed for a well-behaving point-of-care sensing device. Well, I hope I convince you that SYMBIT does comply with all these real-time connectivity, easy specimen collection, and so on. And we are now very much into the physics and the chemistry of the amplification mechanism that we want to understand as well. And I thank all the source of support, so ERC grant, Regione Puglia, the EU Commission for the SYMBIT project, and Regione Lombardia for the BioScreen project, and you for your kind attention.
Thank you, Luisa. Thank you for this exciting but also really talk which goes down deep in what you're doing, and actually a life-saving technology. Are there questions from the audience? Maybe I start one. Out of your experience, how long do you think it will take until we see something of this or used by end users?
We believe we are now at TRL 5. Okay. So, but probably moving very fast into TRL 6, and with the right support, TRL 7 would be very, very close. So we think that we can get to the point where we have an assessed technology that is working out of the lab, and in fact we are now entering the stage of clinical trials with the Regione Puglia, and we are aiming at 1,000 clinical trials. After that, I think we will be very, very beyond TRL 7.
Thank you, Luisa, again for this great talk, and thank you for listening.